Fun fact, many years ago it was found that a patient's smell could be linked to the disease or ailment they suffered from, so people began to try and cure themselves by using perfumes and floral packets to make themselves smell differently. In modern times, we know that the illness is caused by something different, but this has not changed our attraction to scented products. And almost all of us have been guilty at some point of spending far too much money on deliciously scented products, think Lush or The Body Shop. Take, for example, a morning routine where we begin with body wash, shampoo, conditioner, face wash, all containing various forms of scents. Then we move on to hair products, makeup, lotions, potions, perfumes. This is perhaps the most concentrated use of fragrances in a day, but it's far from the only area in which they're used. Consider all the foods and products which also contain fragrances and flavors originally derived or extracted from plants. Over the years, the ways these scents have been extracted have changed, so we've moved from carrying flowers to alcohol distillation, and now we have the ability to extract or synthesize only the precise compound we want. Although chemically there's no difference between a compound extracted from a plant and one synthesized in the lab, the public at large seems to struggle with this, although we as scientists know this isn't true. These days society seems to place great value on natural things. The paleo diet being vegan, anti-vaxxing, organic food. Some of these have more scientific backing than others. Some of them are simply scaremongering by the unintelligent and scientifically illiterate. But what all of them do is play on our inherent fears around what we put in our bodies. This fear is also seen in the strict policy around processing, such as the production of food, cosmetics, and other things we put in and on our bodies. A good example is the fuss kicked up around GMOs, but putting that to one side, policymakers strongly disincentivize the development of new methods of synthesis by their requirements for labeling. Only purely nature-derived materials are allowed to be labeled natural, so there is an incentive for companies to avoid synthesizing nature-identical compounds. The huge inefficiencies in the production of many of these compounds, and growing, harvesting, transporting, storing, processing, and disposing of waste, means that good industrial synthetic methods could be hugely beneficial for both the world and the companies involved in the production of these products. This needs to be identified and addressed by policymakers before this craze for natural destroys nature itself. Even purely synthetic methods may not be the answer. Such methods often involve harsh reaction conditions, petrochemical or otherwise difficult to source starting materials, and huge amounts of waste. The waste produced can be particularly problematic when considering compounds usually sourced from nature. In addition to the usual solvent and extraneous reagent waste, there's typically a large amount of isomeric waste because these compounds typically contain a chiral center. When a chiral center is present in a molecule, conventional synthesis usually forms a racemic mixture of 50-50 of each. In biological systems, the chirality of the compound plays a huge part, which is why the plus isomer of limonene smells like orange, while the minus isomer smells like turpentine, so don't get them mixed up. Where only one isomer in the racemic mixture can be utilized, 50% of it is wasted or needs to somehow be recovered and reconverted so it can be used again. Biocatalysis can offer a solution to many of these problems. The use of biocatalysts for producing nature-identical or even synthetic fragrances and flavors helps the processes to align far better with the 12 principles of green chemistry.
Throughout time, lipases have served important roles in human practices like yogurt and cheese fermentation. But recently, lipases are being exploited as a cheap and versatile catalyst for many reactions in both organic and aqueous mediums. Lipases are found everywhere in nature and are produced by various plants, animals, and microorganisms. However, for the production of industrial enzymes, microorganisms are the preferred source. This is because using animal lipases include presence of trypsin in pig pancreatic lipases, which results in bitter-tasting amino acids and the presence of residual animal hormones or viruses. Plant lipases are also available, but not exploited commercially because of the low yield generated and the processes involved. Thus, microbial lipases are currently receiving more attention because of their technical and economic advantages, where the organisms are cultivated in medium containing appropriate nutrient compositions under controlled conditions. As a biocatalyst, Lipases show great chemioselectivity, regioselectivity, and angioselectivity toward a broad range of substrates. It doesn't require cofactors, nor catalyze side reactions, and remain enzymatically active in organic solvents, with great advantages for the dissolution of the substrates and the recovery of the final product. Lipase is mostly used in mild reaction conditions like room temperature and atmospheric pressure. Also, no specific reaction apparatus is needed. After most reactions, lipases can be recovered and used again. A great number of key flavor and fragrance compounds are found in nature as trace components, and for some compounds, extraction is not a realistic approach, so only synthetic or microbiological pathways allow for their production. For example, menthol is an organic compound made either synthetically or obtained from natural sources like corn mint, peppermint, or other mint oils. Menthol production from natural sources actually result in consumption of more fossil fuels, produces more CO2 effluent, and has more environmental impact than either of the main synthetic routes. This is because in order to produce the same crop year to year, it is necessary to use fertilizers to replenish the nitrogen minerals which are taken up by the plant. Also, there is only 50% of menthol of the dry weight of the herb, so to produce any economic return, it is necessary to use efficient mechanical methods of cultivation and harvesting. Industrially, menthol is manufactured as a single enantiomer by 94% EE on the scale of 3,000 tons per year. Currently, it is mostly synthetically produced, with there being two main reactions used, the Taga-Saigo and the Harman and Reimer processes. This reaction shows the Taga-Saigo process, which uses asymmetric synthesis and the key step of its process. Myrcene is converted into diethyl-anilamine, which is isomerized to plus citronellal in the presence of a chiral rhodium phosphine catalyst. The transformation of citronellal to menthol is performed through cyclization to isoploigal followed by hydrogenation. This reaction shows the Hyman and Reimer process, which starts off with M-cresol reacting with propene to produce thymol. The catalytic hydrogenation of the latter compound gives a mixture of the 8 isomers of menthol. The following fractional distillation affords racemic menthol, which is 2 isomers, and is then converted into the corresponding benzoate. Saponification of the obtained crystalline material affords minus menthol, whereas the mother liquor gives plus menthol. The latter compound and the other 6 isomers are recycled in a separate step. In terms of lipase as a biocatalyst for menthol production, 2 new processes have been recently proposed. Hyman and Reimer were the first, and accomplished the resolution of racemic menthol benzoate by lipase-mediated angioselective hydrolysis to provide minus menthol with almost complete angioselectivity. The other process developed by AECI Limited starts directly from the mixture of the 8 isomers of menthol. The angio and distereoselective acylation of this mixture using lipase yields methyl acetate and 96% EE. The ester is separated from the unreacted isomers by distillation and then hydrolysis to yield minus menthol. In both processes, the undesired isomers are recycled by isomerization.